Welcome to this Matooke product profile presentation at this RTBfoods annual meeting. My name is Elizabeth Hakasa on behalf of the Matooke team. Our presentation is going to follow this outline. We are going to give you information from the field surveys. We shall tell you how we prioritize our characteristics for dissection. We shall go through proofs of concept, developing nearest-based high-throughput selection tools, phenotyping, and 2, the planned activities for period 4. These are our survey results from the field. These are the field data was collected by a multidisciplinary team led by a group of social scientists and gender scientists. And these are the results we got. We spoke to farmers, traders, processors, and consumers. And these are their preferred characteristics. They are actually ranked in order of preference. So the farmers preferred big fingers, big bunches. Traders preferred big fingers, big bunches. And the consumers preferred a Matooke that has a good texture, Matooke aroma, a yellow color, and of course a good taste of Matooke. We went ahead to quantify the characteristics because we wanted to understand what do the end users mean by they want a big bunch. Like you can see initially, the farmers say they want a big bunch, they want big fingers, but what is a big bunch and what are big fingers? So when we went ahead to do this with them, and we found that anything that is below 15 kilograms is considered a small bunch. So what they consider as a big bunch and what they prefer is 15 kilograms and above. For finger sizes, anything that is below 12 centimeters is considered a short finger, and anything that is above 12, that is what they would prefer. We have published our findings in the International Journal of Food Science and Technology, and our main highlights in this paper also bring out the main drivers of preference as a big bunch and big fruits, then also soft texture, good taste and good aroma, plus yellow food. What also comes out in this paper is that the women and the men mention same characteristics of preference, although they rank them differently. We have gone ahead to develop a Matooke sensory trait dictionary, and in this dictionary we give you the traits that we've used and the description for each of the traits. We have used it also has categorical scales, and we have used it to generate data, which data we have already given to the ontology group. From the initial slide that I showed you, the end users, who are the consumers, gave us characteristics ranked in this order: texture, taste, and aroma, and color as their preferred characteristics. They are ranked in that order. But for this, for the work that we have done, we have prioritized 2 of them for the lab, and that is the texture and color. And the analysis we are doing on them are the starch, cell wall, pectins, and rheology. And then for the color, these are the analysis that we are doing. What you see in black here is what they prefer: soft texture, soft pulp. And what you see in red is the bad traits. They don't like this. What you see in green here is the analysis that we are doing on the different preferred prioritized traits. We wanted to understand the optimum level of intensity of the characteristics they selected, so we did a jar. So when you look at mouthfeel, color, and softness, here let's start with mouthfeel. You can see that the intensity of the respondents was so high on Chibuzi here and Nachitembe. So this means that for mouthfeel, they prefer anything that is close to Nachitembe here and Chibuzi. We used 4 cultivars: Chibuzi, Nachitembe, M30, and Narita. And whatever we are doing, our range runs from very good to very bad. So we consider the majority of the consumers want their mouthfeel to be close or just as Nachitembe and Chibuzi. When we come to color here, we see still Chibuzi and Nachitembe taking the lead. They want their color to be just as right as Chibuzi and Nachitembe, and same applies to softness. That is why now we refer to this as our reference cultivars, because their color their mouthfeel, and their softness is just what the consumers would prefer. These are the analyses that we have done in the lab, and these are the methods that we have used to do the analysis. We performed some correlations, and these are sensory texture descriptors and physical parameters. And we can see from this correlation circle that the sensory texture descriptors, these ones here, these are sensory texture descriptors. You can see they are negatively correlated to some physical chemical parameters. We can see dry matter here, instrumental hardness, and we have starch, we have pectin. So these ones are negatively correlated to these ones. But then we can see that also that these parameters are positively correlated to these. These are sensory texture descriptors, hardness and firmness. So you can see that pectin, percent pectin content, total starch is positively correlated with hardness and firmness by touch and by mouth. The biplot also shows us that this shows us that where some cultivars lie, and we can see that this N with a number is a hybrid, and then the rest of the varieties are land races. So we can see that there's a number of hybrids that are on this side that are high, that are very hard, that are high in dry matter intensity and total carbohydrate and total starch. And these, so that means that these parameters in the first and second, these cultivars in the first and second quadrant are hard. So they are positively correlated to the physical chemical parameters here and the sensory parameters. Then the cultivars we see this side have a high intensity, they are moldable, they are smooth, they are very moist, but then these ones are negatively correlated to the physical chemical parameters this side. So that means that the varieties this side are soft, they are not hard, they have low dry matter content. We also did correlations on sensory color parameters. These are sensory color parameters and biochemical parameters. We have total carotenoids content here, flavonoids content, polyphenol content, and total tannins content here. From this, we can see that total carotenoids content is positively correlated with the yellowness of the Matooke and the uniformity of the Matooke. And we can see that total tannins content is negatively correlated to the yellowness. So that means that Matooke that has high tannin content will definitely not be yellow. And this is what is not preferred by the end users. So, and also that means that varieties that have a high polyphenol content are going to have less yellowness and less uniformity of the color. The bioplot also here shows us where the cultivars lie in the different quadrants. In phenotyping, we have done 55 genotypes ranging from best to worst. For all the work that we have done, we do varieties that we consider very good, and we follow a range to what we consider very bad Matooke. And we've been able to collect spectra on raw cooked samples. We have RVA data, and so far we've done 63% of the material. Then what is remaining is we need to evaluate 181 genotypes in work package 4, and we are going to work with a training population for genomic selection. Work package 4 requires a tool to phenotype the population. We are not yet, we haven't developed the tool yet. We've done participatory evaluation in 4 agroecological zones of Uganda. We have sites in 4 of them. That's the Western, Central, Midwest, and East. And on station, we've so far done 65 genotypes. So far, we can see that preferred size, shape, and appearance can directly be used to select at even earlier levels. Then the characteristics can also be used to update the breeders on selection profiles. We communicate our results through publications in journals, inter-program and inter-package meetings, and also through reports. Our key activities for period 4 are going to be publishing the results from work package 1, activities 4 and 5. We shall optimize the soaps. We will also continue with the physicochemical analysis. That's these ones here. And we'll conduct interpackage meetings for Matooke and other product profiles. Then we'll submit the data to PMU. We acknowledge the contribution of the following partners in doing this work. Thank you very much.